Welcome back to the Deep Dive. Today we're diving into one of the most fundamental questions in cancer biology. It's really the origin story of the tumor itself. And we're going to focus on hepatocellular carcinoma, or HCC, liver cancer.
It's a really critical area because HCC, it often shows up in a liver that's already been through a lot.
Been through a lot is putting it mildly. We're talking about chronic injury from viruses, from alcohol, from overnutrition leading to fatty liver disease.
Right, and that constant damage and regeneration cycle sets the stage for a real biological mystery.
And that's our mission for this deep dive. We're tackling a paradox. A person with severe liver disease, their liver is it's full of cells that already have cancer-driving mutations.
Exactly. These somatic mutations, they form these little nodules, millions of them. The liver is just packed with these pre-malignant, genetically altered cells.
That's the mystery, isn't it? If a cirrhotic liver has millions of these little nodules, these ticking time bombs, why is actual HCC still relatively rare in comparison?
It's like having a field full of landmines, but only a few ever go off. The mutation itself, clearly, isn't enough to cause cancer.
So there has to be something else, some kind of biological switch. And the research we're looking at today suggests the switch isn't in the cell, it's all about the cell's address, its neighborhood.
That's right. The key concept, and we're going to keep coming back to this, is metabolic zonation. It's the environment, and it seems to dictate everything.
Okay, let's unpack that. Metabolic zonation. I think of the liver as the body's main chemical factory. So what does zonation mean here?
It means the factory isn't uniform. Imagine a single functional unit of the liver, a lobule, as a sort of circular assembly line. Blood flows in from the outside edge, that's the portal triads, and moves inward toward a central vein in the middle.
And the different locations on that line have different jobs.
Profoundly different. The cells on the outer edge, which we call zone 1, get the blood first. It's fresh, it's full of oxygen. So they specialize in things like making glucose, heavy-duty synthesis.
And by the time the blood gets to the center,
by the time it reaches the cells near the central vein, in zone 3, the oxygen is low. The blood is now carrying waste, toxins. All the stuff zone 1 couldn't handle. So zone 3 cells specialize in detoxification.
So zone 1 is like the pristine front office, and zone 3 is the hazardous waste disposal unit at the back of the factory.
That is a perfect analogy. It's a lower oxygen, higher stress, higher waste environment. And this deep dive is all about showing how that simple address, zone 1 versus zone 3, completely changes what happens to a cell with a cancer mutation.
Okay, let's get into the experiment. The researchers used two very common genetic drivers in human HCC.
Yes, Ctnnb1, which is a major oncogene, and they paired it with a knockout of a tumor suppressor called Arid2. They're often found together in human tumors, so it's a very relevant model.
And to test this idea of location, they created these very clever mouse models where they could turn on the exact same mutations, but in different zones.
Exactly. One group of mice got the mutations only in zone 1, near the portal triads, the front office. And the other group got them only in zone 3, near the central veins, the hazmat unit.
And they had a way to track these cells, right? Some kind of marker.
A brilliant one. They used a protein called glutamine synthetase, or GS.
GS, okay.
Yeah, and normally GS is only found in this tiny little ring of cells, maybe 2 or 3 cells deep, right around the central vein in zone 3. But the Ctnnb1 mutation, it causes GS to pop up everywhere else, ectopically.
So it becomes a bright red flag. You see GS outside that normal ring, you know you're looking at a mutant cell.
A perfect lineage marker. You can visually track every single mutant clone over months.
So the setup is perfect, same genetics, different zip code. Now, let's get to the first mind-bending result. What happened to these mutant cells in their neighborhoods over 6 months?
Well, they were polar opposites. Let's start with the zone 1 mice, the ones in the nice neighborhood. The number of mutant cells, those GS-positive cells, it was stable. They formed these clusters and they grew.
They were competitive.
Very competitive. When they looked at which cells were actively dividing, they found that over 65% of all proliferating cells in that area were the mutant ones.
Wow. So they're not just surviving, they're taking over. They are fitter than the healthy cells around them.
That's the definition of high clonal fitness. They established a thriving colony. Now, contrast that with zone 3.
The tough neighborhood.
The tough neighborhood.
Yeah.
Here, the number of mutant cells actually went down over the 6 months. They were being outcompeted by the healthy cells.
They were disappearing.
They were disappearing. Only about 30% of the dividing cells in that zone were mutants. They were struggling to survive, being actively purged.
Okay, so let's do a quick recap here. This is a massive contrast.
You have the exact same mutation. In zone 1, it's a super competitor. It expands, it dominates. In zone 3, it's a loser. It struggles, it gets pushed out. So based on everything we know about cancer, you would bet all your money that the tumors will come from zone 1, right?
Yeah.
The expanding clone.
You bet the house on it.
So for you listening, what do you think actually happened? Because here's the plot twist.
The results just west. They shatter that assumption. Despite all that growth in zone 1, the tumors overwhelmingly started in zone 3.
That is the central paradox. The zone where the mutant cells were thriving was mostly safe. The zone where they were dying off, that was the cancer hotspot. The numbers are just staggering.
They are. So 80% of the mice eventually developed HCC, but the bias was so clear. A full 90% of the zone 3 mice developed at least one visible tumor.
90%. And in the zone 1 mice?
Only 59%. And the tumor burden, the number of tumors per liver, it was almost 3 times higher in zone 3. An average of 9.6 tumors versus just 3.6 in zone 1.
Okay, let me just repeat that. Nearly 3 times the number of tumors in the area where the precancerous cells were literally being outcompeted and disappearing.
Right. So the conclusion is unavoidable. The amount of clonal expansion, how much the mutant cells grow before becoming cancer, it's irrelevant.
It doesn't predict risk.
Not at all. The risk is being set by something external. Something in the zone 3 environment that is helping these struggling cells make that final leap into malignancy.
So they had to go on a hunt for these transformation accelerators in zone 3. How did they find them?
They did a big genetic screen. They compared all the genes that are expressed in healthy zone 1 versus healthy zone 3 cells and picked out the ones that were most different. They ended up with 11 candidates.
And they needed a fast way to test them.
Right. They used a technique called hydrodynamic transfection. It's a way to quickly blast oncogenes right into the mouse liver and cause tumors to form very, very fast. It's like a rapid assay for cancer susceptibility.
And out of that screen, what popped?
2 genes stood out, Gstm2 and Gstm3.
Gstm, what are they?
They're members of the glutathione S-transferase family, GSTMs for short. And when they deleted these genes, HCC development it stalled. The mice survived much longer.
Okay, so GSTMs are the accelerators. What is their normal job in the cell?
They're phase 2 detoxification enzymes. Their home base is zone 3 because that's the part of the liver that sees the most toxins and metabolic waste.
So they're the cleanup crew we talked about.
The specialized cleanup crew, exactly. Their job is to grab onto harmful chemicals and neutralize them by sticking a glutathione molecule on them. It makes them water-soluble so the body can get rid of them. They are the frontline defense for the most stressed-out zone in the liver.
This is starting to click. Zone 3 is a dangerous place, so it has this incredibly strong built-in defense system. And now it seems like the cancer cells are hijacking it.
They're co-opting it. And this isn't just in mice. In human HCC patients, having high levels of Gstm3 is linked to shorter survival. This is a real human mechanism.
But they still needed to prove that Gstm was acting on the environment to kickstart the cancer, not just helping an already formed tumor grow.
The final definitive experiment, they went back to the zone 1 mutant mice, the ones that almost never got tumors. And they just temporarily turned on extra Gstm3 in zone 1.
You're kidding. They just added the accelerator to the safe zone.
That's all they did. And the result was stunning. It completely shifted the tumor origin. In the control mice, only about 22% of tumors started in zone 1. But when they added Gstm3 to zone 1, that number shot up to 61%.
So recap 2. The Gstm proteins, which are a normal part of the zone 3 environment, act as a transformation accelerator. It's not about how fast the mutant cells grow, but whether this accelerator is present. But that leaves a big question. How does an enzyme that's meant to protect a cell from damage actually help it become cancerous?
It protects the cell from a very specific kind of death, a process called ferroptosis.
Ferroptosis. Okay, we need to define that one very clearly.
It's different from apoptosis, which is kind of a clean, programmed cell suicide. Ferroptosis is, the name gives it away, ferro for iron. It's death by fire and rust.
Death by rust.
It's driven by uncontrolled lipid peroxidation. The fatty components of the cell's membrane start to oxidize, to go rancid, fueled by iron. The cell literally rusts from the inside out and then bursts. It's a catastrophic death.
And because zone 3 is so high in oxidative stress,
its cells are constantly living on the edge of ferroptosis. So when a mutant cell pops up there, it's under immense pressure to die this way. But it hijacks the Gstm defense system.
The Gstms act as an anti-ferroptosis shield.
A powerful one. They're not helping the cell divide faster, they're just helping it not die. They're giving it this survival advantage to hang on long enough to acquire more mutations and transform.
And they proved this in the lab.
Absolutely. When they deleted Gstm3, the premalignant cells showed all the signs of oxidative stress and started dying more. And in cancer cell lines, if you delete Gstm3, the cells become incredibly sensitive to drugs that specifically trigger ferroptosis.
Which means you found a vulnerability, a therapeutic target. If cancer initiation depends on this shield, can you take the shield away with a drug?
You can. They tested a drug called ethacrynic acid. It's actually an old loop diuretic, but it's known to inhibit this family of GSTM proteins.
And did it work in the mice?
It worked beautifully. Treating the mice with ethacrynic acid dramatically reduced the tumor burden. It was almost as good as genetically deleting the gene. Wow.
And they confirmed the mechanism. The drug made cancer cells vulnerable to ferroptosis, but if you then added a different drug that blocks ferroptosis, a chemical called Ctnnb1, the cancer cells were saved.
A fire extinguisher for the rest.
Exactly. It proves the drug is working by taking away that anti-ferroptosis shield. And they did one last experiment to really hammer it home.
What was that?
They used another anti-ferroptosis enzyme called FSP1. Think of it as a backup shield. They took mice where they had deleted Gstm3 so tumor growth was stopped, and then they turned on this backup shield, FSP1.
And the tumors came back.
The tumors came roaring back. It proves beyond a shadow of a doubt that Gstm3's only important job here is to stop ferroptosis in those early vulnerable precancerous cells.
And this isn't just about this one specific mutation, is it? It's a general rule.
It seems to be. They tested other cancer models, including one caused by a chemical carcinogen. They all showed the same pattern. Tumors start in zone 3, and they depend on Gstm3 to get going. This appears to be a universal gateway for liver cancer initiation. This has been an incredible deep dive. We've completely flipped the script. We learned that in the liver, how fast your premalignant clone grows is nowhere near as important as where it lives. Zone 1 cells are fitter, but zone 3 is where the danger is.
Because zone 3 cells learn to survive in a hostile environment by co-opting these powerful defense systems, the GSTMs, to protect themselves from that fiery death of ferroptosis. That protection is what gives them the time to become fully malignant.
The clinical implication is huge. We're now looking at a very specific, environment-dependent bottleneck that we can target. Instead of carpet bombing a tumor that's already formed, maybe we can prevent it from ever starting.
Exactly. Using drugs like ethacrynic acid could be a zonation-dependent strategy to help high-risk patients.
Okay, so for our final provocative thought, a question for you to mull over. The research shows that these GSTM enzymes are essential. They protect the healthy, normal zone 3 cells from dying in that harsh environment every single day.
That's the problem.
Right. So if a drug like ethacrynic acid broadly inhibits this protective system, acting on both healthy cells and the rare mutant ones, how do you design a future therapy that selectively pushes only the premalignant cells into ferroptosis without causing massive collateral damage to the healthy liver tissue that you desperately need to keep?
Finding that therapeutic window, exploiting the cancer's addiction to a defense system without crippling the body's own defenses, that is the next great challenge this research presents.
A fascinating problem to solve. Thank you for joining us for this deep dive into the origins of HCC. We hope you feel thoroughly well informed.